Hi, my name is Michael, and I'll be doing my presentation today where I investigated the characteristics of biodiversity metrics as well as tested them on published biodiversity data. To start with, one aspect of managing biodiversity that is important is knowing what is in a particular site you are managing. There are many measurements that can be used to obtain this information, first of which is the species richness. This is a measurement of the number of species that are in a particular area. While this information is useful and easy to obtain, there are some issues with it. For instance, look at these two populations. They both have the same species richness, but despite this, you can tell that there are other characteristics that make these two populations different or unique. There are other measurements that take this into account, including the Simpson's diversity and Shannon diversity. And what these measurements do is they take into account the abundance or the number of individuals in a population, as well as the evenness, which is a measurement of how similar the abundance between species is. While these measurements are also useful, there are also some issues with them. To demonstrate this, I made a little community here, and the important thing to notice about this community is that as you continue to increase the sample size, the species richness also increases. This is because species richness is dependent on sample size. So it makes it difficult to know whether you have enough samples for your species richness estimate to be accurate. To get over this, people use the asymptote. And so this is when the species richness starts to level off as the sample size increases. And once this happens, you can be confident that your species richness is a accurate measure. But this brings into another problem in comparing two different communities that have different sample sizes. Because the community with the bigger sample size will often have the bigger species richness. To get over this, people have started creating rarefaction curve, which plot the number of species as a function of these samples. And this is something we should be familiar with as we have created these before in lab. The problem with the ones we created in lab, though, is that R mathematically calculates the rarefaction curve. And while it gives you a nice smooth line, nature isn't often this predictable, and there's often a lot of variation within the measurements. And so by doing rarefaction curves this way, you run the risk of overestimating the actual species richness. So the objective of my study then is to create a function that can create a rarefaction curve that takes into account this variability. I also want to use this function to determine specific characteristics about these biodiversity indices. And then finally, I want to test my function on actual published biodiversity data. So to start with, I would like to give you an overview of my function. And so what it does is it samples without replacement multiple times for the total number of individuals in a data set. And so to give you an idea of how this works, I created a very simple abundance data in the top left corner. And in this, they have a total of 5 individuals. And so what my function does, it will take that 5 individuals, then resample them for a sample size of 1, and then it would go again and then resample them again for a sample size of 2. And then you do this again until you get the total number of individuals. And so in this case, 5. So 5 resampling. And so because of this, you got 5 new data sets that all have different abundances. And so my function then takes each of these and then recalculates the richness as well as the Shannon and Simpson diversity for each sample size. And so with a sample size of 1, you can see you got a richness of 1 and a diversity index of 1 as well. And then you resample, and this time you have 2 species. And so by doing this, as you increase the sample size, you have more variation within the data set. And to give you a better idea of how my function works, I looked at the data from lab 3 again. And so in the original data set, you can see that the blue and the green curve look pretty distinct. However, when you run it through my function and you add that variability, it doesn't seem to appear that we could be confident that these two different populations have a different richness. And so my function just kind of adds a little more information to the rarefaction curve. And so the next part of my project was to use the function to determine specific characteristics of the biodiversity indices. And so the first thing I wanted to look at is how evenness affected the indices. And to do this, I created 5 separate populations that each varied in evenness slightly. I did control the number of species within each population as well as the species richness. So the only thing that varies between these 5 populations is the evenness. And so after doing that and running it through the function, the thing that you most notice for the species richness is that the more even the population is, the faster the curve asymptotes, and you can be confident in the richness. Whereas the more uneven the population, the longer or more samples you need before that curve starts to asymptote. Further, if you look at how it impacts the diversity indices, first thing you notice is that the more even the population is, the higher the diversity, and the less even, the lower the diversity. And this is similar for both Simpson and the Shannon diversity. But something other interesting to notice is that the Shannon diversity always has a slightly higher diversity metric than the Simpsons does. And this is because the Simpsons diversity puts a higher weight on the abundance or evenness of a species, or not species, population, than the Shannon diversity does. And so the next factor I wanted to look at is I wanted to see how sample size impacted these biodiversity metrics. And to make sure it was only the sample size that was changed, I made sure the new population data I used had a very similar structure to the previous one, so we can know it's only the sample size that is causing any changes to the data set. And so if you look at species richness, this is the previous species richness I used at the smaller sample size, and this is the one with the bigger sample size. And so the structure of the species richness is very similar, so not much change. Maybe the only difference is you'll be more confident if you obtain your richness at a higher sample size. And this is also the case for the diversity. So increasing the sample size by itself does not appear to impact these indices on its own. But this is not a fair way to measure an increase in sample size because, as mentioned earlier, as you increase the number of samples, you're more likely to encounter more species, so your species richness is also likely to increase. So it's also important to see how species richness is going to impact these biodiversity metrics. And so to do this, I created another set of populations, but this time I controlled for the evenness and the number of individuals. And so each population has a varying number of species. And so once you run this through the function, you could tell that, first of all, of course, if you have more species, you're going to have a higher species richness. But what I find more interesting is where all the curves asymptote. And when you have a higher richness, you also asymptote at a higher number of samples. So you need a greater sample to be able to be confident in your richness calculation. And this is also the case for both the diversity indices, whereas if you have a lower number of species, you asymptote at a lower rate and need a less of a sample size to be confident in your estimate. And so what I gathered from this mini investigation is that if you increase the evenness of your population, you don't actually decrease the species richness, but what you do decrease is the number of species, or not number of species, number of samples needed to be able to be confident in your species richness calculation and for the species richness to asymptote. Also, as you increase the evenness, you increase the calculation you get for the Shannon diversity, and this is also the case for the Simpsons diversity, but because the Simpsons diversity has more weight on the abundance of these species, it will not increase at the same rate as the Shannon diversity. Also, if you increase the sample size by itself and nothing else changes, you're not going to see a difference. But if the increase in sample size is also associated with a shift in the population dynamics in some way, all 3 of the indices would see an increase. And so after doing that, the next part of my project was to see how my function held up against real life published biodiversity data. And so I went out and I found this study that had the species abundance data available as well for me to analyze. And so what this study looked at was empty forest. And so what they wanted to see is whether the nature reserves within China actually acted as refuges for the species in the area, or if it was another case of a forest devoid of diversity. And so to do this, they had 3 separate land categories. The first was pristine habitat, which were the nature reserves. degraded habitats, which were the surrounding forests that weren't protected, and then they had open areas, which was considered farmland. And so they wanted to see if there was a difference in the richness between these three land categories. And so they came out and they published data that looked like this. And so quickly break down the chart. The black bars are the total species found in that particular habitat type. The gray bars are the species that are widely dispersed that were found in that habitat type, and then the white bars are the species that are more localized that do not have that big of a range. And so this is how they presented the data, and it doesn't really tell you much about the richness. First of all, it doesn't show if the species richness has asymptote at all. So I don't know if this richness is an accurate representation of the richness of the area. Further, to get the average richness, what they did is they took the richness from each different plot that they sampled and then averaged it together. And because they're comparing the richness between a bunch of separate sites, I don't know if they took into account how the sampling effect would impact the richness and if each site had a similar sample size. And so it's hard to tell what this chart is telling you. And so I went through and I wanted to determine this for them. And so what I looked at was the black bar, all the species in total, because they didn't really tell you which species were considered widely dispersed or localized. So I couldn't really tell you the difference between the gray and the white bar, but I could present the data on the black bars. And so this is the results that I got from running it through my function. And so I broke it down into the three separate habitat types as well as they did. But what you can tell, first of all, is that it does seem to plateau at a pretty quick rate for all three of them. I don't know if we'd be confident in the very low sample size because some of the plots had a low sample size. So I would prefer if they had a higher sample size, but it doesn't look like it would be a significant increase in richness if they do increase the sample size. Further, between the three plots, it doesn't look like there is a major difference in the species richness. So in the original study, they concluded that because there wasn't a great difference in richness between the three sites, you can consider the pristine forest or the nature reserves as empty forest and an empty habitat type that doesn't actually increase the diversity. And after running it through my function, it looks like they did get the correct conclusion. They just kind of got it going the wrong way. So even though they did get the right conclusion, I believe there are better ways they could have gone about presenting their data. And the first thing they could have done is to not use bar charts. Bar charts do not provide enough information for the reader to make any inferences about the actual nature of the richness of your population. And so it's better to use rarefaction curves because it gives the reader a better idea of what the actual species richness of your site is. Further, if you're going to publish biodiversity data, published the abundance as well as the richness. There was multiple times where I was going through a published journal and I'll go through their data and they only had the species richness, and that doesn't provide enough information to be able to do any further analysis on that data. And further, because you only post in the species richness, it immediately makes me question whether or not your diversity data is accurate from the start. So you kind of lose some credibility. So if you're going to publish the abundance data with it. And so anyways, I'd like you to thank you for listening. If you have any questions, please email me, and I'll respond to you if I don't die from the coronavirus first. So anyways, thank you for listening. And here are my references as well.